Should we go ahead and get started?
Yeah, sounds good.
Okay, welcome to the third and final day of the Land Model and BGC working group meetings. So today we have 5 talks in the morning, and then followed by the CGD seminar by Rosalie Fisher, and then this Cupid tutorial in the afternoon. And then we, to warn you all, there's going to be a lot of children here today, 200 children. And so, yeah, the Damon room is reserved for us for lunch in case you can't find place in the cafeteria. So just so you're aware. Yes. Yeah, and then I'm the first speaker, so.
Yeah, so I'm going to introduce Kristen, who's going to give us updates on ocean biology.
All right, thank you, Gretchen. Okay, so hello everybody. As Gretchen just said, I'm going to talk about the different things that are coming into CSM3 with regards to ocean biogeochemistry, and I'd just like to acknowledge my collaborators here who have helped me with this, put together this talk. This is a quick overview about what I'll talk about, some of the new developments for CSM3 MARBLE, which is the marine biogeochemistry model for CSM3. We're having a new planktonic ecosystem, and then we'll have explicit coccolithophores, which I'll talk about all of these in more detail, and then fully variable phytoplankton stoichiometry, and some new iron cycle updates, as well as potential solution to the overly extensive oxygen minimum zones that were in CSM2. So just to get started, here is, so for CSM3, we'll have a 4 phytoplankton, 2 zooplankton version of MARBLE. And if you all remember, I don't know if you knew this, but for CSM3, you had only 3 phytoplankton and 1 generic zooplankton type. And so we're getting slightly more complicated here. We'll have 4 phytoplankton types. And on this diagram that I made, those are on the top row. And then the small phytoplankton and the diatom circles are slightly larger because they really comprise the bulk of primary productivity in the ocean in the model. But the diazotrophs and coccolithophores have important biogeochemical roles. Diazotrophs can fix nitrogen, and coccolithophores do calcification, and I'll talk about them in more detail in the next few slides. And then you'll notice on the bottom row are the 2 zooplankton types. So rather than having 1 generic zooplankton, we're splitting that into 2. So the small ones, the microzooplankton, the larger ones are the mesozooplankton. And this having this kind of separation allows us to couple with an offline fish model. And that's what I'm showing here. This is a diagram showing how the CSM MARBLE connects to the feisty fish model. And so you can see that the mesozooplankton type is used as a kind of a food for the fish in the model, the small size class of the fish. And so feisty is a really cool model that has 3 fish functional types, and you have different size classes of each type, and fish can grow from one size class to the next. That's represented by the orange arrows on this diagram. And then when they get to the largest size class and they have enough energy, they can allocate some of that towards reproduction and produce more of the small size class of fish. So it's a really nice model, and I put the food web down there in the lower right corner. And so next I'll talk about the coccolithophore functional type that's going to be in CSM3. So coccolithophores are kind of calcifying phytoplankton, and they make these beautiful calcium carbonate shells. And I've put some of my illustrations along the left-hand side of the screen. And then the background is a image from NASA of a coccolithophore bloom in the North Atlantic. And so coccolithophores are really important to the carbon cycle for several reasons. One is that by doing calcification, they remove alkalinity from the ocean, the surface ocean, and that affects air CO2 uptake. And then also they make this calcium carbonate shell, and that's heavier than organic matter, and that can ballast or pull or help sink organic matter to the deep ocean, where that carbon can be sequestered from the atmosphere for quite some time. And then their ocean acidification with the absorption of anthropogenic CO2 into the ocean can affect their growth and their calcification. And so in MARBLE, we modeled 2 parts of the coccolithophore biomass. One is the particulate inorganic carbon shell, so that's the calcium carbonate or chalk shell. And then the other part is their organic carbon biomass, so that's like the guts inside of the cell there. And so the ratio of these, the PIC to POC or the PIC-POC ratio is like a measure of the degree to which the coccolithophores are calcified. And so as CO2 aqueous CO2 increases in the ocean, so ocean acidification, then PIC-POC tends to decrease. And we put these relationships into Marble, and they're based on a large meta-analysis of many laboratory experiments for different species of coccolithophores. And so in general, if we go forward in time, you have more ocean acidification. That means less calcification, more alkalinity staying in the surface ocean. So potentially it can absorb more CO2, but then you also have a less calcium carbonate shells ballasting organic matter to the deep sea. So next I'll talk about the fully variable phytoplankton stoichiometry that's going to be in CSM3. So CSM2 also had variable phytoplankton stoichiometry, but not a nitrogen to carbon ratios. That was a fixed ratio. So now that is also fully variable. So now I put a map here of a small phytoplankton nitrogen to carbon ratio in a MOM6 Marble development run. And so you can see how that varies with the ocean. So the whole parameterization of phytoplankton stoichiometry has been kind of revamped for this version of Marble. And each phytoplankton type has a minimum and a maximum, an element to carbon ratios. And so for this small phytoplankton nitrogen to carbon, we have the lowest ratios in the places where nitrogen is limiting. Those are all the purple areas on this map. And then the higher ratios in where nitrogen is plentiful, like the Southern Ocean. And this allows us to better match particulate organic matter stoichiometry. So this plot is showing the 100-meter POC flux iron to carbon ratio as a function of dissolved iron concentration. And so the previous version of the model is in red, the observations are in blue, and now the new version with the tuned stoichiometry is in the black dots. So they're better matching the observations. And this under climate change, you could get more expansion of nutrient limited areas. And so this could have important implications for the biological carbon pump. So next I'll talk about a few iron updates, and this is really high level. I'm not going to go into a lot of the details, but iron is a really important control on phytoplankton growth in many regions. And this map here shows the most limiting nutrient for diatoms in a MOM6 Marble development run. And all the areas in this orangish yellow color here are iron limited. So that iron availability is really controlling the productivity in those regions. And iron comes from many sources. All of these have been updated for CSM3. One of the most important ones is the iron from dust and black carbon deposition from the atmosphere. We also have iron from rivers, iron coming in from sediments, and iron from hydrothermal vents. And I'll go to a little bit more details on this iron from dust and black carbon. And so on this top plot, we have soluble percentage, so how much of that iron can actually dissolve into the ocean. And this is a function of the coarse to fine dust or black carbon ratio. And so as you are really close to source regions, there's a high coarse to fine ratio. And so somewhere like that would be like just the dust coming off the Sahara, for example. And not a lot of that is actually very soluble. But as you get more distant from the source regions, a lot of that coarse dust settles out, and you get a higher coarse to fine ratio, or a lower coarse to fine ratio, and much more of that is soluble. So somewhere like that would be like the equatorial Pacific right there, which is distant from many sources, but it has a higher solubility. Another new update that I wanted to inform you all about is this new parameterization in MOM6 called Lethe backscatter that helps the overly extensive oxygen minimum zones in the model. And so these are plots of the zonal jets as a function of depth and latitude here. And you can see that without this parameterization, you don't get these jets that are actually seen in observations, which are on this side of the slide. And they're called Tuchia jets, and they actually really help to oxygenate this part of the ocean. And so So these maps along the top are oxygen at 500 meters, and I put a contour there as a 20 millimole per meter cubed oxygen concentration contour, and that's kind of the threshold for an OMZ. And we can see that before adding the backscatter, it's the oxygen minimum zone is getting really big. This is more like what was in CSM2, but and then the observations from World Ocean Atlas are in the middle. And CSM with the backscatter parameterization is on the right. So we can see that this is really helping that big OMZ. It's not perfect, but it's a big improvement. And you can see that in these bias maps here too, a big bias, negative bias there. And then it's improved with the backscatter. And then this not only helps oxygen, but it also helps nitrate, and actually even more. And so on this, these are plots of RMSE as a function of or the root mean square error, and oxygen on the top as a function of depth, and nitrogen on the bottom, the error on the bottom for nitrate. And the blue line is without backscatter, and then you can see the improvement in oxygen, so less error with the backscatter parameterization. And then if you look at nitrate, it's actually even a bigger improvement. We had a big reduction in the nitrate error, and this is because when there's low oxygen, that triggers denitrification, which is the remineralization of the organic matter. That is, instead of consuming oxygen, it consumes nitrate. And so it leads to a big negative bias in nitrate if you don't have enough oxygen. And then I just wanted to touch on two kind of aspirational plans for CSM3 with regard to ocean biogeochemistry. And one is this even more complicated version of MARBLE with 8 phytoplankton types and 4 zooplankton types. And this was already developed for CSM2, but we're hoping to get it into CSM3 as well. And then lastly, the ocean biogeochemistry coupling with the sea ice. So that's basically having sea ice algae. And then the sea ice and the ocean can actually exchange nutrients and carbon and things like that. So that's all.
I don't know if it means we've got actually a fair amount of time for questions. And Katie.
Thanks, Kristen. I'm wondering how you prioritize the specific types of phytoplankton and zooplankton. Like you mentioned at the end there, a more complex model. Are there trade-offs with like what you want to represent versus like the complexity there?
Yeah. So every phytoplankton type has, I think now 5 tracers associated with it because we have variable stoichiometry. So every one of those elements is tracked in the ocean. So it is like a significant computational cost to add like a phytoplankton type. So in the one that we're actually using for the model or for CSM3, the 4P2Z version, that one, the diatom group and the small phytoplankton group are actually representing a whole bunch of different species, so they're very generalized. And then, but then it's really important to have the diazotrophs and the coprolithophores because they do important BGT things. So we have to consider all of those things in terms of how much complexity we want to represent for this workhorse version of CSM3, but having the biogeochemistry taken care of, as well as you can answer important ecological questions with the 2 zooplankton types in combination with the fish model. So we would definitely want 2 zooplankton. Plus, having 2 zooplankton allows more mixed phytoplankton communities, which is having those 2 top holes and the mesozooplankton grazes and the microzooplankton. So having that more complex food web than the 3P1Z version that was in CSM2 is allowing more realistic communities to come out.
Not that it's a competition, but what is the complexity level of MARBLE relative to, or do you know relative to some of the other Earth system models that are doing ocean BGC? Is it?
The 4P2Z version is pretty comparable to someone like Cobalt has. I think I have 3 zooplankton types actually. So, and we have versions that have more zooplankton, but generally, we don't need that extra zooplankton to answer science questions. So I didn't think it was, necessary for the workhorse version of the model. And so, but yeah, I think it's actually pretty comparable, at least to the ones I know. But I don't know about the CMIP7s because I haven't looked at what the other modeling centers are doing besides GFDL. And it's about the same. but they don't have explicit coccolithophora's. It's just a kind of fraction of the small phytoplankton type.
Do you know, is marble used in the NORESM or just, because they have a Nemo ocean, right? I think, and then so.
They don't participate in our marble developer meetings.
Yeah, maybe it's probably not.
They have their own ocean BGC model. I forget its name, but their whole ocean and ocean BGC is completely separate from CESM, unlike the other parts of the model.
I want to say first, I love your illustrations. And for those of you who don't know, she does all of her own illustrations and everything. But I guess what I'm wondering is about the phytoplankton and zooplankton. Are there spatial distribution? Like how is it determined where they are? Are they just everywhere in all oceans all the time?
So there's always a little bit of every type that we have like a minimum amount, so they can grow anywhere that conditions are favorable. Now, light, temperature, and nutrient status actually control how abundant they are. And then, diatoms do better under low light, high nutrient conditions. Small phytoplankton are more set up for nutrient limited areas like warmer, like the oligotrophic gyres in the ocean. Coccolithophoras are forming blooms in the high latitude spring or in summer months, and diazotrophs are kind of stuck in the subtropics, and they do their nitrogen fixation in places where nitrogen is limiting, because that's where they have the competitive advantage. So they kind of just pop up where they're supposed to most of the time. Sometimes they don't behave. I'm trying to tune them for CSM3. So.
Any more questions? All right, if not, we will move on to Holly Alvarez.
Okay, how's that sound?
All right, good morning. Thank you for having me. I am a NOAA climate and global change postdoc fellow here at UCAR, and my mentor is Natalie Freeman at NOAA PSL here in Boulder. So I'm going to talk about solar radiation management and future ocean carbon uptake. The intention behind solar radiation management is to limit the amount of solar radiation reaching Earth's surface while society finds the means to cut CO2 emissions and scrub CO2 from the atmosphere to achieve a more stable climate. Today, I'm going to focus on one proposed strategy that's in the modeling stage. Stratospheric aerosol injections, or SAI, is currently the most popular proposed intervention, and it uses volcanic eruptions as an analog. This cartoon from SRM 360 helps us visualize how SRM could be deployed. It consists of approximately 12 metric tons of sulfur dioxide being injected into the stratosphere from planes. And the simulations that I'm using for this project are referred to as the ARISE simulations. I've placed here how we get to the acronym, but for the sake of time, I'm not going to read it. But I do want to point out that the research community does agree that climate interventions are not a replacement for the elimination of fossil fuels. And I think it's important to say that in any conversations that we're having about climate interventions, because I'm already hearing people say, oh, well, then we can just keep doing what we're doing. But no, this is a temporary solution, as I mentioned, as we're working to stabilize our climate system. And then also I want to say SAI is not the solution, and it's definitely not the only solution. And again, we're in the research phases. This figure is showing global mean temperature from 2020 to 2070, with temperature in kelvins along the x-axis and pre-industrial temperature above pre-industrial in kelvins on the right axis. And the red lines represent SSP 2-4.5, which I'll discuss in a minute. And the blue lines, which run from the year 2035 and wrap up in 2070, are the ARISE SAI 1.5 simulations. And they work to maintain a temperature at or near 1.5 degrees Celsius above the pre-industrial value. There are 6 ensemble members each for the SAI forced runs and for the control SSP 2-4.5 runs. The reason I specify that is the SSP 2-4.5 runs are connected with WACOM 6 to make them comparable to the ARISE simulation or the SAI simulation. And then we use the ensemble mean of each for our analysis as an estimate of the forced SAI response. And so in this figure, Richter et al. Were able to show their success in achieving their goal with the simulation. And so I'm now using these simulations to study the impact on ocean biogeochemistry, such as how SAI could impact the ocean as a carbon sink. The simulations use the middle of the road shared socioeconomic pathway emission scenario that represents a future where greenhouse gas emissions continue, but then start declining mid-century. And that leads to roughly 2.7 degrees Celsius warming by the year 2100. So the acronym, I know you all have heard it, but SSP 2-4.5. So on the figure on the right, it's circled there in green. Okay, so now that I've reviewed the simulations, I'll get talking about the ocean carbon sink. In recent history, air-to-sea CO2 flux trends look something like this. CO2 is not absorbed everywhere, absorbed the same everywhere. Here, the mid-latitudes are blue, representing carbon going into the ocean, where areas of red, like the eastern equatorial Pacific, show carbon going out of the ocean. So this is kind of a baseline of recent trends that I use as we look at SAI simulations into the future. And so I'm going to show you some preliminary plots of the future. So looking broadly at the ARISE simulations of air-to-sea CO2 flux, here I'm showing the decadal mean for the present decade on the far left, the mean for the SAI experiment years, 2035 to 2070 in the middle, and then the difference between the two on the far right. The units here are moles of carbon per square meter per year, and overall the ocean is a carbon sink through this period. So looking at the far left and the middle plots, purple areas represent carbon going into the ocean, green represents carbon coming out of the ocean, and the patterns here match what I showed on the last slide. But looking at the differences between the two over on the right, the average flux from deploying SAI for 35 years compared to the present decade shows mostly blue, representing an ocean carbon sink, with some slight pale reds, and then some strong reds in the Arctic and the North Atlantic there at the start of the Great Conveyor Belt. The blues represent increased carbon uptake, particularly in the high latitude Southern Ocean. But we do see declined uptake in the North Pacific and North Atlantic and the Arctic, and some in the subpolar regions compared to the present decade. So this was just kind of my start to look at, 35 years versus the present, overall, what are we looking at? So then I broke it down into decades, so that we can look at decadal means compared to the present decadal mean. And also I wanted to see the change over time. So here again, the red areas represent declined uptake, while the blue areas represent increased uptake. And the short version is that globally, the ocean as a carbon sink persists under SAI, but with some questions about what would happen with additional time. So first, looking left to right at the plots, the carbon uptake in the Southern Ocean, do you see my mouse? Yeah. The carbon uptake in the Southern Ocean here persists, but the subpolar regions, in all of the basins and in the Arctic, we see declined uptake with time. That's where we see these pale reds compared to during the current decade. And by the 2060s, the North Atlantic shows substantially reduced uptake. So looking at the impacts of air-to-sea flux this way, I would be really curious to see if these impacts would persist and or amplify if the simulations ran longer than 2070. Over more than the 35-year simulation, how would carbon uptake change? That's my personal big question here. So my plan going forward is to look at things on basin and regional scales. And I'm thinking of ways to show where changes in flux reflect changes in internal variability, such as physical circulation, climate patterns, biological activity. And of course, pair this with discussion of the overall function and stability of the ocean carbon sink. I guess I'll say it here, but my audience in doing this work and getting published manuscripts, I feel, is for the SRM community, specifically the SAI community, because there at present are not really any published works about ocean biogeochemistry, the impacts of SAI on those. And there's a lot that have been published about land and atmosphere. There's one on marine ecosystems that just came out that's fantastic, but we need more of a voice for the ocean before we're taking any sort of action. And there are some researchers that are calling for small-scale deployment experiments to begin. And I personally am concerned because we don't have this work published yet. So I put that out there because today I'm asking for input and ideas. I want to educate the SAI researchers in a way that they can contextualize what's happening in the ocean with their work, knowing that they're not ocean specialists. So one more results slide, I'm going to show a time series of the rate of change in air-to-sea CO2 flux from 2015 to 2100. So along the x-axis is time, and then using the SSP 2-4.5 as the control run in purple, or I've labeled it the if we do nothing curve, compared to if we deploy SAI in red. Carbon is now in petagrams per year as a rate, and the more negative the values get down the y-axis indicates more carbon going into the ocean. So overall, we can contribute SAI to increasing the ocean carbon sink by approximately 0.13 petagrams of carbon per year over 35 years in the model. And if I wrote my paper that way, we could say, oh, SAI is a positive for the ocean as a carbon sink. But I feel concerned when I look at this figure this way, because it appears that the carbon uptake does not persist with time. And so I think this gives interesting insight and context as to the efficacy of SAI. And please help me understand if you interpret this differently. But to me, it appears that SAI simulations, if they were run longer in the model, it's possible that SAI might only temporarily increase the amount of carbon absorbed by the ocean before it trends back to the if we do nothing curve after the year 2070. So here, and even the start of the simulation, the end of the simulation aren't too far off from the if we do nothing curve. And so, yeah, this is kind of where I'm looking at how do I tell this story, because we want to be objective and not say, yeah, we should do SAI or no, we shouldn't do SAI. But at the same time, when we have folks that are kind of already in motion about SAI, I want to kind of say, wait, what about the little guy, the ocean? So that's important to me. It's on my mind a lot. And then one more thing before I finish up, because I work in the theoretical climate model space, I always like to talk about the real world work. Approximately 40% of Earth's human population depends on marine life. And as I perform these experiments, in my mind, the real work is to create space for Indigenous and other researchers to be involved now while we're in the research stage, as we can consider ways to achieve a more stable climate. And I'm always looking for collaborators, ideas, so feel free to reach out to me anytime. And last slides, in summary, SAI enhances the ocean as a carbon sink for the years studied, but many questions regarding long-term impacts, not only globally, but regionally as well. My goal is to have this manuscript ready for submission by the end of June, and I'll be presenting it this summer at the Gordon Conference, which is focused on climate intervention. So that'll give me a good opportunity to get in dialogue with a lot of the SAI researchers there. All right, thank you for your time and attention.
Okay, we've got time for questions. Keith.
Hi, Holly. Nice to see you. Very nice talk. You said one of the motivations is that this is a stopgap until we reduce emissions for this sort of approach. Are there any experiments in the SAI group of experiments where they stop doing the SAI to, and you might see, does the ocean burp out that extra carbon that it in that meantime absorbed?
Yes, there are experiments, modeled experiments that show that if they were to stop SAI abruptly, we would absolutely have a reversing effect and maybe even worse than it was before. So that's definitely a consideration. And so I think I did put an extra slide in here. This came from Zarnetsky et al., where they were looking at marine ecosystem impacts. The idea was the black curve, if we're cutting emissions and we're doing CO2 removal, then the solar radiation intervention would be the blue arrows. So the idea is we ramp up solar radiation intervention for SAI as we are organizing ourselves for a large-scale CO2 removal. And then we're phasing out SAI as CO2 removal has really taken place on a large scale. That's kind of the logic. And so it hopefully wouldn't be an abrupt stopping. It would, in theory, happen in parallel with our CO2 removal taking place.
I had a very similar question about how long that carbon would stay. So,
Peter?
So this is great. We actually have run a rise out of 2100 for one of the ensemble members. So we have that. But we've also, in our climate interventions work, been looking at using CAM6 with the stratospheric aerosols from WACM, from the ARISE simulations. We've got a 5-member ensemble of that running to 2100 with emission driven. So we've got the CO2 in the atmosphere also like evolving as it's running. Really happy to share those with you if you'd like to have a look. We're also doing exactly this. We're doing CDR and then working out how to do the peak shaving.
Oh, wow.
So we've got simulations for that as well.
We need to talk.
We do. And we've talked before, but following this up.
Yeah, thank you.
This is really brilliant.
Yeah, thanks.
Yeah, on the time series plot where you have the difference between the two, the with and without stratospheric aerosols, that you integrate that difference in the ocean sinks and calculate using like a TCRE or something, like what the equivalent temperature change would be. How does that like additional marginal temperature change to kind of due to this enhanced ocean uptake compare to the actual stratospheric aerosol quality?
That's an interesting question. I think the way that the way that the simulations are set up, this doesn't really show it, but is that it's I do mention it here. So the way that the simulations are set up is that SAI is injected once per year. So at the end, there's an algorithm. They call it a controller algorithm. So it decides at the end of each year, okay, how much SO2 do we need for the upcoming year so that we can keep meeting that 1.5 degree C mark. And so I don't, in that format, I don't see the temperature change that I could gauge it that way.
Well, I guess you've got, because you're doing concentration driven, that then whatever the climate effect from that CO2 uptake, enhanced CO2 uptake of the ocean isn't expressed in the actual climate. So you could consider that as like an additional cooling. So if you know how much extra carbon the ocean is taking up, you could then think of that as like additional to whatever. I mean, I guess you kind of know how much cooling you've done stratospherically because you can compare that to the without stratospheric aerosol.
Yeah.
Right, but then you could compare that temperature difference versus whatever would result if it had been an emissions driven run from the extra increment of ocean carbon uptake.
Okay. I'm trying to wrap my brain around that. I don't really think of, maybe somebody can correct me if I'm wrong, but I don't really think of extra carbon going into the ocean impacting the atmospheric temperature.
It wouldn't in a concentration driven run, but it would in an emission driven run.
Okay, sure. Okay. So
It is of the order of about 1 gigaton of CO2. So if you think about it, it's going to be less than 0.1 of a degree
okay.
in terms of the carbon cycle on top of the SAI.
Yeah. That's a good question. I hadn't been thinking of it that way. I'll keep that in mind.
I know we probably need to move on, but maybe just is this ocean acidification part of the story? It seems like that's
Very much so.
Yes.
Yeah, this is my second postdoc project. My first one is on my first one is on aragonite saturation state. and exactly showing SAI does nothing for ocean acidification. Again, if you just look at the period studied, it looks like it decreases, or basically it slows down ocean acidification slightly. But when you put it in the grand scheme of things, it's just not even touching it in relation to what we would need. So that's the big red flag that's coming up first in a manuscript.
All right, thanks for the great discussion and good fun. Next up, we have Katie Rosie. She's going to be talking about land program responses.
Can folks hear me okay? Yeah, great. Okay, so head back to the land. And normally there's not that many soil biogeochemistry talks at these things, but there was quite a few yesterday, so that makes me very excited. Yeah, for those who know me, I'm Katie Rossi. I'm currently a postdoc at University of California, Merced, but was recently a postdoc at CU and at Michigan and a long-term visitor here at NCAR working on this project. Okay, so even though we've talked a bit about soil biogeochemistry, I still like to remind everybody why it matters and why it might be important for understanding carbon cycle responses to climate change. And I always like to use this paper from Cesar Terra a couple of years ago. And so basically what this work shows is that soil biogeochemical mechanisms matter. So here's an example of a soil biogeochemical mechanism, whether a plant has arbuscular ectomycorrhizal fungi associated with it. You can maybe argue this is a plant mechanism, but it affects the soil too. And now we're looking at the effect of elevated carbon dioxide on the carbon pools. And what you can see is that depending on the fungal association, you have these different responses, right? And at least for this specific mechanism, it's not very well presented in models currently. So this is now showing you the effect on plants on the x-axis and the effect on soil on the y-axis. And you can see the observations show this kind of trade-off with plants and with soils, but the models really don't show this. And so this is just one example where we might be able to better represent soil biogeochemical mechanisms. Unfortunately, CLM6 has two different types of soil biogeochemical models we can use. And we heard a little bit about this yesterday. So kind of what I call the default model is CLM Century. It's a century-like model, which is kind of this classic understanding of soil decomposition that we put in our models. And then we also can use CLM with mimics. This model developed by Will, now over 10 years ago. And if you were at Will's talk yesterday, you heard a bit about this model, but some kind of key features it has is that it represents microbial community and physiology, and so we can look at those mechanisms. And then it also allows us to decouple soil and plant responses a bit because of those microbes kind of in the middle of the plants and the soil. And so we can use these different models. We might assume that these different models produce different responses to climate change. So when we think about looking at climate change and observations, we have to think about these big global change experiments. So this is one called TerraCon. That stands for, well, I'll get that in a second actually, but it's a global change experiment initiated in a tall grass prairie in Minnesota in 2012. And the TerraCon stands for temperature. So that's these heat lamps facing the ground. There's also below ground heating in the soil. There's these rainfall reduction tents, I guess, that people have to go out and put out when it's raining. There's these elevated CO2 pipes. So these pipes just pipe in more CO2 to these areas. And then they fertilize every year. So they have these 4 global changes they're representing. And we're going to focus on temperature and elevated CO2. And importantly, we're going to focus on their interaction. And I point this out because these experiments are really rare. There's like maybe 2 to 4 in the globe that do this many global changes at once, all interactively. They're very expensive. They're hard to maintain. And so there's a suggestion that if we can represent individual global changes in our models well, we'll also be able to represent the interactions well. But we're not really sure about that. We haven't really evaluated that. And so that's another goal of this work. Okay. So what do? We set up the model to look like the site. So we have a mix of C3 and C4 plants. We use leaf seed and SLA from the site. And then this is a super, super sandy site with low organic matter. So we change the soil to look like that. Then we calibrate CLM Sentry to the control. So we spin up with just 2020. That's because Mimics only needs 1 year of spin up. So we tested that, made sure that looked okay. We do 200 years of AD and post AD, and then we're using PPE tools that Linnea helped me modify for our site. And then for Mimics, we have to do this Anderson acceleration spin up, so a different kind of spin up. And then we end up doing hand tuning. So we can't quite figure, or I haven't figured out how to code the PPE tools in with Anderson acceleration at least. Okay. So then once we have a calibrated model, we'll run the model under global changes. And we're trying to match the experiment as closely as possible. And so in this case, we're doing elevated CO2 as zeta, which is a plus 180 ppm. And then unfortunately, I have not made folks who were at the CLM meeting a few weeks ago remember this, but I haven't quite figured out how to warm the soils yet. So far just air warming, but we're figuring that out. And then we'll evaluate that against the observations. And then the final step is to think about some, what I'm calling these ad hoc parameterizations, and I'll explain that a little bit when we get there. But what other changes can we make to the models inform my observations to make the model look more like the observations? And this could potentially suggest future mechanistic changes we could make. Okay. So calibration. So calibrating to land carbon pools in the control situation. I have the same plant parameters for both models. If you want to look at those, they're on the screen. Happy to talk more about those. And then different soil parameters because the models have different parameters for Sentry and for Mimics. Okay, and so this is what these calibration plots look like. So just the observed on the x-axis there, you can see the high variability from year to year at the site, and then the modeled on the y-axis, and then that's the one-to-one line, so we're trying to get closer to there. The default is in blue and the calibrated orange. So basically, we are able to calibrate the models. We really struggled with this originally. And so you can see there's a slight trade-off here between slightly underestimated plant carbon in Mimics and overestimated Sentry. So we had a really hard time growing plants in Mimics. Highlighting that Mimics is quite nitrogen limited, which we're aware of from some previous work. Okay, so we have calibrated model, and then we can look at these global change effects. Okay, so here is shoot carbon, the observed results for shoot carbon at the experiment. And so what you can see is that shoot carbon increases under warming, elevated CO2, and then under both kind of additively increases, meaning that warming and elevated CO2 responses added together are pretty comparable to the interactive effect. But what do we see in our models? So for the Sentry version of CLM, we see a pretty good result. It looks relatively similar. You could maybe say the warming is a little bit underestimated. And then again, remember, we're expecting that Mimics is going to look pretty different, right? Because it's a different model. But actually we find that Mimics looks very similar to Sentry in when we put those in CLM. And so that's kind of one outcome of these results. Okay, moving on to root carbon. We see in the observations that root carbon looks a little bit different than shoot carbon in terms of it has more of an interactive effect of warming and elevated CO2 together. So you can see that although root carbon increases with warming and elevated CO2 individually, when you put these together, you get a weaker response. So that's an interactive or synergistic effect. And then if we look at the model responses, I'm going to show both at once because they, one, look similar again, but two, look really similar to the above ground. So below ground and above ground plants look very similar in their response to global change here. Okay, and then finally the soil. So soil responses and the observations are weaker, as you can see, which is not surprising, right? The soil generally responds less strongly than plants. But you can also see that there's this slight increase with warming and elevated CO2, and then really no response when you can put them together. So this really strong synergistic effect. Okay, if we look at the models. So for Sentry, CLM Sentry, we are getting elevated CO2 looking right in terms of the directionality of the response. But the interactions, once again, showing these very additive effects, and then there's really no response to warming. For Mimics, it's a similar story, except we'd see a slight loss of carbon under warming for Mimics. And so takeaways from these kind of observation model comparisons. In general, the model's responses were weaker in terms of like a mean % change than the observed responses. The observations generally had more positive responses to warming than the model did. And then the observations also had more interactive responses than the models. And so the next step of this was then to see, are there mechanisms we could change in the models to make them look more like the observations, to potentially inform like future model development. Okay, so I don't expect you to read this table. Let me say that up front because nobody wants to read a table like this in a presentation. But what we did is a bunch of ad hoc parameter changes, I'm calling it. So they're basically individual parameterizations for each global change. So they couldn't really be applied directly, but just to give us an idea of what mechanisms might be important. And then we're looking at a bunch of kind of ecological drivers that are inspired by either site data that we have from Cedar Creek or previous research. And I want to point out that some of these mechanisms can only be applied to the CLM with mimics because some of them involve microbial physiology. So this is kind of a cool thing we can do with mimics. Okay, so I tested a bunch of these, but I'm just going to show you the best ones. So in this case, for Century, the two changes that led to the model looking more like the observations were changing the allocation between roots and shoots. And then also having higher litter decomposition, which allows for more nitrogen mineralization for the plants to grow. Both of those with warming. For mimics, we find different drivers being important. We find actually allocation changes the same. So changing that root to shoot ratio is important also for mimics, which might kind of indicate like something we can really look at and changing in the model to get these global change responses looking better. But the other thing we changed for mimics that worked out well is the lower cost of arbuscular mycorrhizal nitrogen acquisition. And this is because other work at the site has shown that there's higher total below ground carbon allocation with warming, which tends to either mean like more root exudation, more mycorrhizal uptake, which allows for more nitrogen for the plants to get more nitrogen. And then finally, higher microbial turnover was also found to be important, which many articles have found to be a common response to higher warming. Okay, so what do these look like with these ad hoc parameter changes? There's a lot to look at, but basically what I want you to take away is that really if you focus on the warming responses in yellow here, these are more positive and stronger, which just looks more like our observations. Okay, so overall takeaways. First, we can't necessarily presume that our models are going to give us insight about combined global changes if they're getting individual global changes right. And so we need to think more about these interactions and these synergies and how we can represent those better. I think a lot of times when we think about putting microbes in models, we're like, they're going to make everything look better. And in this case, putting microbes in the model didn't necessarily provide a better looking response of carbon pool responses to global change. But having a representation of microbes in the models does allow for us to look at these mechanisms that we can't look at without the microbes in the model. And so I think that's like an exciting thing we can look at. And that's all I have. Thanks.
I think we've got a couple of minutes for questions.
You described the changes as ad hoc. I guess it's just, they're ad hoc in the sense that like, I guess what you could have done alternatively would like recalibrate for the response, right?
Sure.
And then well, my question, would you maybe have gotten the same parameter change or parameter changes if you had done like a calibration to the response? And then you would call that not ad hoc, or parameter changes is something we do all the time. So I guess
I guess what I mean by ad hoc is like for the allocation example, for example, like that seems like something that's like maybe going to happen. And so you could create a function with temperature and allocation or something. And that's more right. It's more sustainable than like a one-off parameter change.
so okay. So it's like in place of what should be a parameterization, you just did a parameter change.
Sure. Yeah.
Yeah, I guess I wanted to clarify. So I was asking, like, so yeah, the parameter changes, they're not so much like a combination, like the parameter values should be these now. It's more like we need to implement a process, like it's more like an acclimation process. Is that maybe what you're?
Yeah, these are great questions.
No, because
part of the project side.
No, because like like Linnea was talking about like calibrating like the land, like the veg model to like trends. But this seems like this could be another interesting way to calibrate like other parts of CLM is like we want to like there are all these experiments. Like, can we like do this throughout the whole model and like be able to turn on soil warming and then like but that begs the question, like, do we need a parameter change, like a state change, or is it that we need to represent some acclimation process that's going on that?
Yeah, I guess I've been thinking about it as more of like a process you implement some sort of like temperature effect on allocation, on mycorrhizal nitrogen uptake or something. But yeah, maybe you could just calibrate with a local change in the control together and you get something different.
Yeah. But you might break the model under current conditions. if it is an acclimation process that you're not representing, then you would break it under the current conditions.
Yeah. I think we'll have to save the rest of the discussion for after the last talk. So we'll move on to Lily Wu, who is hopefully online. Lily, are you here?
Yes, I'm here.
Do you want to share your screen?
Yes, I will go ahead and do that. Okay, I think it's sharing now. Can everybody see? Okay, great. Okay, can everybody see the screen? Yes, okay, awesome. All right, well, hi everybody, my name is Lily. I am a second year PhD student at Cornell University. And today I'm gonna be talking about my research modeling volcanic ash impacts of the 2010 Eyjafjallajökull eruption. So we're shifting gears a little bit and looking at volcanoes. So why do we care about volcanic ash? You can see from the schematic I made, there's lots of different impacts we can really talk about. We can focus on the societal impacts. So that's mainly looking at the disruption to aviation and human health. But we can also look at some radiative impacts and also potential chemistry interactions with other volcanic emissions like sulfates. And then we can also look at the land biogeochemistry and ocean biogeochemistry impacts. Of course, for this presentation, I'm going to be focusing on ocean biogeochemistry. So really looking at the role volcanic ash plays into that. And so we're focusing on the soluble iron that comes from the volcanic ash and how that connects to ocean biogeochemistry. So we saw from the previous slide, volcanic ash has lots of important impacts on the climate and Earth systems, but volcanic ash was previously neglected in these large-scale global climate models like CESM in favor of volcanic sulfate aerosols. And this is because ash particles are larger and assumed to be shorter-lived, so their impact would be limited to smaller scales, both temporally and spatially, again, compared to these volcanic sulfate emissions. So the goal of this research is to incorporate a new volcanic ash tracer into CESM to first simulate a volcanic eruption and then analyze the ash impacts. And so to first simulate our volcano, we are going to be using a case study. And the one I'm using is the 2010 Eyjafjallajökull eruption in Iceland. The explosive part of this eruption occurred from April 14th to May 17th. And we also had 4 eruptive phases with different ash chemistry. So we can see in this image right here, we have Iceland, and then at the southern tip, that's where the volcano is located. And we can see this massive ash plume that's just coming out of the volcano. So really, this volcano added a lot of ash to the atmosphere. This is also a really good case study to use because we have observations of the eruption for both the emissions to constrain our model inputs, and then we also have observations of the impact on Europe, so we can validate the model as well. All right, and then just very briefly, talking about the model setup. So I'm using CSM 2.1 with the community atmosphere model, or CAM6. So I'm coming at this from an atmospheric perspective rather than like a land or ocean perspective. We're also using the modal aerosol model, or MAM4. So this includes 3 different aerosol sizes. We have the Aitken accumulation and the coarse mode aerosol sizes. And then we also have the mechanism with intermediate complexity for modeling iron, or MIMI. And MIMI is especially useful for modeling the iron aerosols that are going to be coming out of the volcanic ash. So how do we incorporate volcanic ash into the model? Here we're building upon the existing dust framework in the model. So we're assuming that volcanic ash is behaving similarly to mineral dust. We have an extended version of the model that carries 8 dust tracers, but a previous sensitivity study showed that we don't need all 8 to actually capture the radiative forcing of the dust. So we're just going to condense those dust tracers into 4, and then we're going to use the remaining 4 tracers as our new ash tracers. And these are divided based on the mineralogy from the IFL local ash observations in Pack et al. And so this table right here is just showing how we're dividing up each of those ash tracers. And so a percentage of the mass will go into each tracer. And again, these are divided based on composition. And the percentage of mass will also vary in time, depending on what phase of the eruption we're in as well. And then in addition to dividing up all those tracers, we also ran a bunch of sensitivity studies because some of these volcanic parameters are poorly known. So these include things like the vertical distribution of the ash in the plume height, the particle size distribution of the ash, and the deposition velocity or the shape of the ash. So the first 3, the low, base, and the high, these are going to be changing the plume height distribution. So for the low, we're assuming a constant mass mixing ratio throughout the entire ash plume. For the base, we're assuming 90% of the mass is in the top kilometer and then the rest is evenly in the bottom. And then for the high, we're assuming 100% of the mass is at the maximum plume height. We also have some small sensitivity studies. So these are assuming more mass in the smaller accumulation aerosol size bins. And then we have some depth sensitivity studies as well, which are increasing the deposition velocity to kind of account for the shape of the volcanic ash. So this panel right here at the top is a visual representation of those plume height distribution sensitivity studies that I just talked about. So we have the altitude in the y-axis in kilometers right here. So if we look at the colors, again, the green is the low or the constant mass mixing ratio sensitivity study. The orange is the base and the blue is the high. And so this is just one snapshot in time, but this vertical profile will vary in time depending on what the actual plume height of the eruption was. And so here in the background, we have the time series of that from April to May. And so the blue line in the time series represents our plume height in kilometers. And then the brown line represents the eruptive mass in teragrams. And so we can get these plume height and eruptive mass from remote sensing observations. So that's kind of all of the input information we need into the model. And so once we have all of that, we can throw everything into the model and simulate our volcanic eruption. So now I'll just show some results. So these next couple of results have observations to compare to help validate our model. The first result I'm showing here is the particulate matter or PM10. So not really related to ocean biogeochemistry, but it's important to analyze PM10 because we want to understand how well the model is capturing the long-range transport of the volcanic ash. And so if we focus on the spatial plot, panel A right here, we can see Iceland in the middle, and then Europe is in the bottom right. And so the model is the contours underneath, and then each of these circles represent a different observation location. And so these are station observations taking PM10 measurements, and these come from the AeroMap data set. Panel B right here is showing the scatter plot of those. So we have the observation values on the x-axis, and then the model values on the y-axis. And then panel C is showing the correlation coefficients for each of our different sensitivity studies. And then the purple color right here represents a background case without any volcanic ash added to the model. So if we focus on panel C, we can see that the model is performing relatively poorly compared to the background case without any volcanic ash. And so we think this is because of model spatial resolution issues, especially further away. So this really suggests that the model is not capturing the long-range transport of the volcanic ash very well. But what about closer to the volcano? So we're switching gears a little bit, and now we're looking at the soluble iron concentration at the surface. And so we're focusing on soluble iron because of its potential to impact ocean biogeochemistry. And so previous studies have shown that volcanic ash could be a significant source of soluble iron. So that's why we're focusing on this. And so if we look at the spatial plot, again, the model is the contours underneath, and then our circles represent our different observation locations. And these observations are coming from the RRS Discovery, which is a research cruise that happened to be in the area during the eruption. And so they were able to measure some of these concentrations at the surface. And so panel B is showing the time series of those observations in black. And then the green line represents one of our model sensitivity study values. Panel C, again, is showing the correlation coefficients for each of our different sensitivity studies. And then the purple is the background case without any volcanic ash added to the model. And so again, we can see that the model is performing relatively poorly compared to a background case without any volcanic ash. And we think especially very close to the volcano, there just seems to be a 3-day timing delay that I'm not showing here. But when we kind of adjust for that timing offset, we're able to better capture the observed values. So this really suggests that at least very close to the volcano, we can capture the magnitude of the observations, but there just seems to be a 3-day timing delay, possibly be because of model spatial resolution issues or interpolation issues very close to the volcano. Okay, and then I'll show one last result. So here we're looking at the soluble iron deposition. And so we don't have observations to compare this to, but it's really important to understand the deposition for the potential Earth system impacts. So here we're really trying to understand how much more soluble iron did the volcanic ash add to the North Atlantic Ocean ecosystem. And so if we focus on panel B, on the y-axis, I have each of our different sensitivity studies, and then the y-axis is X times more. So just how much more iron is the volcanic ash adding to the ocean compared to a background case without any volcanic ash. And so if we look at the numbers here, on the lower end, we have about 24 times more soluble iron that's being added. And on the higher end, we have about 45 times more soluble iron that's being added. And so this is really just a lot of soluble iron. We can see even the calculated mass I have in gigagrams. So really just a lot of soluble iron that's coming from the volcanic ash. And again, this has potential downstream impacts on ocean biogeochemistry as well. Okay, so last slide, wrapping it up. I have lots of conclusions here, but I'll leave you guys on two key takeaways. When we're looking at the unknown volcanic parameters, we find that the ash plume height distribution matters a lot more on the sensitivity of the results compared to the particle size distribution or the deposition velocity. And when we're looking at the soluble iron, we find that the volcanic ash is adding a lot more to the ocean ecosystem over the North Atlantic compared to a background case without any volcanic ash. And so this really has important implications for ocean biogeochemistry downstream. And then overall, we just need more work to be done to better model the volcanic ash. Thank you, and I'll take any questions.
Anybody have any questions for Lily?
So Lily, this is great. I'm wondering if there is a temperature response that is associated with some of this increased ash within the aerosol model. I
haven't looked directly at the temperature, but I did plot like the radiative forcing. That tells us, how much like the energy imbalance is, and we find that locally, very close to the volcano, there's a really strong positive radiative forcing. So this suggests a lot of warming at the surface. So yeah, but I'd be interested in looking at the temperature response, especially like the first couple of days after the eruption.
Yeah, thanks for a great talk. Did you look in the ocean part of the model and see if there's increased primary productivity from the iron deposition after the volcano and some of your simulations?
Yeah, so actually, I don't think they have ocean biogeochemistry turned on because I'm coming at this from like just looking at the atmospheric part. But of course, my next step would be to, try to see if we can couple that and then look at if there is an increase. Because I think some studies have, there's observational results showing that there was an increase. So I'd be interested to see if the model can capture that.
Yeah, that'd be great. Sometimes the North Atlantic is iron limited, but not all the time. So it kind of.
All right, thanks again. Now we have our last talk of the session, which will be Natalie Mahols.
Hopefully, can you see me and hear me?
Yeah. Do you want to share your screen?
Let's see, did that work? Okay, there we go.
Yeah, we see your desktop.
All right, which one are you seeing? Are you seeing the right part?
We see presenter mode.
How's that look?
Good. Yeah, there you go.
Okay, great. All right. Well, thanks so much for having me. I wish I was there. We've basically not seen the sun for about 6 weeks, and I assume you're seeing the sun there. That's normal. Anyway, I want to talk to you about a paper that we submitted to Global Biogeochemical Cycles. You can see that there's 1 billion co-authors on there. And we're taking a look at the model estimates of the atmospheric aluminum, calcium, iron, silicon, and titanium concentrations. And we're using the new emit surface mineralogy. And so that's some of the co-authors, and then some of the co-authors are also people who have observations. So this also represents a first compilation of some of these elements. So the goal number one is to try to understand the atmospheric cycling of the biogeochemically important species, aluminum, calcium. Of course, we've done iron, silica, and titanium. So we're focusing on these elements because they're the dominant elements in dust, along with oxygen. Some of these can be very important for the oceans. For example, iron limitation, blah, blah, blah. We don't need to talk about it, but silica can be important for shell formation, for diatoms, for example. Aluminum can be important, linked to the silica, and maybe the calcium isn't super important in the ocean just because there's lots of calcium in the oceans already, but calcium cycling in the ocean is of course really important for the calcium carbonate. On the land side, often calcium can be an important basic element to counteract acids, and it's also in the atmosphere. Calcium is one of the few basic elements in the atmosphere, and so it can actually neutralize some acids that we throw in the atmosphere. And I guess Danny Leung is actually putting some of this into the model, which is great. So we can, from an atmospheric perspective, see it. We're going to be looking here at results from the EMIT project, which is the Earth Surface Mineral Dust Investigation Project. It's a NASA venture project, $100 million or so. And it was super cool. Imaging spectrometer was put onto the space station, and the way they justified this amazing instrument is because of some of the studies that we had done. A grad student of mine, Rachel Scanza, has put speciated dust within the CESM. And so I was deputy PI on this project because of that. The dust radiative forcing is, of course, really dependent on the dust composition. And the imaging spectrometer gives us information about the dust composition at the surface of the Earth in arid regions. And we input that into the models in the CESM, for example, and we calculate the mineral distributions. And, we also calculate the radiative forcing, et cetera. I'm not going to talk about this here, but I'm going to talk about then what the minerals tell us about the elements. We have been evaluating so our second goal here is to actually try to use the elemental data to help evaluate the emit data. So there is also mineral composition in the atmosphere data. And so Maria Gonzalez-Dedos is working on comparing that to the model results showing our mineralogy from the dust regions. But there's actually not that much data. This is a plot of where the individual observations are, okay? So it's pretty sparse. It's a really hard measurement to make. So in this study, we're taking a look at the elemental data, and there's just more elemental data. It's a much easier measurement to make. Of course, not all this data is super close to the desert regions. And so we want to also blank out the regions that aren't dust dominated here. So the first step is to try to understand what the non-dust sources are of different elements. So here's just an example of the elemental data that's available for aluminum in the PM10 size fraction, just as an example. There's just much more data here. These are annual averages. These are individual observations. There's just many more observations of elemental. So how do we go from minerals to elements? Well, we use measured values that for each of the minerals. Most of these observations are from North Africa, but then we can get the aluminum, calcium, iron, et cetera, concentrations for each of these minerals, and then map them over in the model then. So if we take a look at the budgets for these different elements, here we provide the total atmospheric budget in the PM10 size fraction. And, you can see there's, much more, say, silica, 2 orders of magnitude more than titanium, okay, just because the composition, and most of the sources actually dust, but they have quite different sources in teragrams per year. But the percent contribution for all of these, like these black and gray values are the dust part. And then any color, the green or the purple, those are non-dust sources of each of these elements. So we add in our literature search of other sources. We have a good iron database from Sagar Rathod, one of Tammy Bond's students, put together an iron database, and we use some ratios to try to get from the iron to these other elements. We also add wildfires and volcanic estimates. We actually do the volcanoes kind of stupidly right now, so we're hoping next to extend Lily's project to show, to do a better job, because for some elements, volcanoes are actually fairly important, say for iron here and silica. This light one is the volcano. So not so much for iron, it's more for the silica and the aluminum. It can be important. Then also we have the different elements that indicate different minerals that we're actually detecting from the emit. But there is a little bit of an issue there that we find, because if you take a look, iron can be from iron oxides or illites. So like if we go back to this slide, you can see, the iron oxides are the goethite and the hematite here, and they have a lot of iron, but there's very little of them, maybe 1 or 2% in dust, whereas the illite is maybe 20, 30% of the dust, but it's 4% iron. So it actually kind of convolutes the problem, where if you're taking a look just at the iron, you have a good fraction of it that's from the iron oxides, but a good fraction of it is also from the illite. So it's not a one-to-one relationship all the time here. So then if we start taking a look at the observational and model comparison, here's the, just like Lily was showing, we have the model in the background and the observations on top here, and then we have a scatter plot here, and we're using annual averages and climatological averages in each case, and you can see that the values span 4 orders of magnitude, 5 orders of magnitude, and so we're trying to capture all the high aluminum areas and the low aluminum areas, because these all matter for the biogeochemistry. And the observations are shown on the scatter plot on the left-hand side, and then the model is on the bottom here, and the one-to-one line, and then what our error estimates suggest we should get. The largest source of uncertainty in the comparison of the model to the odds we estimate is actually the spatial variability within a 1 degree by 1 degree grid. It can be quite a bit of variability there. There's also a little bit of variability from trying to capture individual events, because a lot of times the aerosols come in individual events. Anyway, here you can see we do an okay job, I'd say, simulating the aluminum. There's quite a bit outside our error bounds. We expect about 1/3 of the observations to be outside our error bounds, and we might get a little bit more than that. We also, this is the station data, so it's climatological mean land stations. And then we also have some cruise data also. And of course, there's much larger uncertainty since these are daily average values. A lot of the time, I'd say maybe we're on the right planet here, basically, is what we're trying to get. We can do this again for calcium as well. Quite a bit of spread, a little smaller change in how much of calcium you get, just because you have many more other sources of calcium. It's got more important, say, ocean sources or volcanic sources than aluminum. And here's, again, some cruise data. Again, I'd say, okay, maybe we're on the right planet. For iron, as you might expect, since our input data sets are much better, we've been working on iron for 20 years now, we do much better than iron. It's not perfect still, but we do much better for iron. Silica, again, we can show that we're somewhat close. There's not very much silica data on the cruises. And titanium, again, we can see that we're roughly right, even in both areas close to the desert regions as well as in the non-dust regions. So one of the things we calculate then, now we can calculate our non-dust fraction, and it differs by element. So this is the fraction of non-dust. So, calcium has the largest area that's dominated by non-dust elements, and iron has the smallest area dominated by non-dust elements. approximately. So in those regions, of course, you can't use any of these. In the regions that are dominated by non-dust sources, you shouldn't be using any of these elements as proxies for dust. So this is kind of the first attempt to articulate this for most of these elements, except for iron. This is the first time the non-dust parts have been simulated and compared to observations. And so, we really needed to do this to evaluate emit, but also to better understand the atmospheric cycle, because you can see that over most of the planet, the non-dust sources dominate, especially over the oceans. We can also estimate the present day to PI ratios. The PD to PI is the present day to PI ratios. And we're using here then the crocodile estimate that there's a 55% increase in dust, especially in North Africa, that's based on the limited paleoclimate data. We just forced the model to capture that. Of course, then the dust-dominated minerals are going to show at least a 50% increase here. So that's what you're seeing here, that, say the aluminum, the iron, the silica, those are, you see big increases in the regions downwind from North Africa where you have the big change. And calcium much less because it has other sources. So just to conclude, this is the first elements of the aluminum, calcium, and silica and titanium from non-dust sources. So it tells us where these elements can be used as a proxy for dust. And of course, they can be improved. We're working on volcanoes. The way we included it here is really quite dumb, but we have estimates at least. But with Lily's work, then we'll be able to extend that, hopefully. Then, of course, some of these depositions could be important for ocean and land biogeochemistry. So we make these deposition fields available. And there appears to have been, or through our estimates, we say there's an increase in some of these minerals. And we're hoping in the next step, we're hoping to see what information it can provide about what's better about emit than our pre-emit estimates. And we're going to test that much more in an upcoming paper. Thank you.
Thank you, Natalie. Questions? Charlie.
Thanks, Natalie. That was really cool. I guess the last bullet point of like testing against a pre-emit data. So is the idea that you do like a fixed composition as like kind of a null model and ask if the emit has higher prediction than that? Is that the idea?
Yeah, we could do something like that. But also we have pre-emit estimates, and they're based on a compilation, either the Clarkin et al. 1999 or the Journet et al. And basically they're based on about 500 soil samples or a few more observations. And you just say, well, this kind of soil has this composition, and then take the soil maps that are available and extrapolate wildly. And so that's what we could do before. And so we'll use those data sets. And those capture some of the issues we showed in Zhang et al. 2015 that you can do a little bit better for, I can't remember exactly which elements. I think, calcium for sure, maybe iron, one other element. You can do a little bit better if you use that kind of information compared to the observations that we had at that time. Here we have many more observations, and we're hoping to show that emit is doing a better job. Basically, before we were making stuff up and extrapolating wildly, and now we actually have observations of what the mineralogy is in the source area. So it's very cool.
Yeah, Katie.
I guess I'm wondering, given that answer, if you could essentially reverse that, like, could you use the emit data to tell you about the mineralogy of the soil?
That's what we did. Yeah. Because the emit gives you the mineralogy of the soil. Yeah.
Great.
Does that make sense? And then from that, we get the elemental.
Okay, cool. Thank you.
Okay. But we have to make a lot of assumptions there. So.
We can open it up more broadly for discussion about any of the talks in this morning session or connections, what we've seen earlier this week.
Yeah, I guess I could use a little bit of a refresher of which of these things are connected already in the biogeochemistry simulation and which ones still need to be connected. So, is there already a connection, I think, between dust and dust wafting and then dust iron deposition into the ocean for ocean BGC.
Yeah.
Would it be important to connect some of these other things? Or like what's there already and what would be the next things we'd want to think about? Sorry, I'm looking at you, Kristen.
Yeah, so there is the iron from dust and from black carbon. So we have like combustion iron sources as well. And those are basically, I think, the only ones that are in the ocean model, is it?
I think we assume a constant phosphorus content in the dust deposition. So in Natalie's work here with the prognostic fraction would be a step forward in reality. I don't know offhand how much of a role that would play, but that's one thing that is currently in the model of the constituent that she mentioned.
Natalie?
The phosphorus has quite a bit of combustion sources also. So if we really wanted to do it right, then we would add like the black carbon again, except that the phosphorus, you ocean biogeochemists have never showed that the phosphorus matters. So it's not worth coupling, I don't think. But so only if you care.
There's very few places where phosphorus is the limiting nutrient.
Yeah, sometimes in the subtropical North Atlantic, there's enough iron from the dust from Sahara, and then the nitrogen fixers can use that, and then they increase the nitrogen, and then phosphorus can come in because then there's enough iron, or nitrogen from the nitrogen fixing group. So, but yeah, it doesn't always pop up. I know in some of my mom's 6 marble development runs, I'm having a hard time getting the phosphorus limitation in the North Atlantic to show up, but it was in CSM2. But that was the main spots. And then maybe the Mediterranean sometimes.
Yeah, I mean, so it could be relatively easy to use the black carbon similar to that we do with the dust, and add in the combustion sources of phosphorus because they're quite important if you wanted to.
In reality, where does phosphorus matter to the ocean?
Just the North Atlantic,
Oh, that's it.
and then sometimes the Mediterranean as well. And those are the 2 main regions. But there were talk of that it becoming more prominent limitation, even in the North Pacific, with climate change. And I think the warming, stratification, more nitrogen fixation, but that really relies on iron. So that nitrogen fixation is high, like it's the nitrogenase enzyme needs a lot of iron. So if you supply the iron, then you can shift and then that fixes the nitrogen, which is the nitrogen supply, which then can shift in the limiting nutrient function of phosphorus. So it could happen, but it's hard to measure.
My recollection is I think we also do a similar thing for silica. That I think we have a fraction of silica out of the dust. It's just a constant fraction. That might matter more. I think we have that. I haven't looked at that line of code in a while.
Do the volcanic eruptions, the CSFCs, do anything to ocean BGC?
I think they can. Yeah, I haven't explored that myself. Looking at the impact of volcanic eruptions, but actually, Holly, didn't you look at that?
Yeah, that was CSM1, and so
But still,
I was looking at temperature.
Oh, it's not just temperature. Okay.
And we did the same. Actually, we did do CSM2. I didn't do it, but remade the large ensemble without the Mount Pinatubo eruption in 1991, and looked at the ocean biogeochemistry that way. But again, we weren't. None of that had anything to do with what was in the atmosphere falling to the ocean.
You guys get to do so much cool biogeochemistry.
Well, you guys too.
We're stuck with the 2-element biogeochemistry.
Oh, okay.
Can I ask also about recap of the plans to get an ocean biogeochemistry spin up for CSM3? Sorry to ask that question, but I feel like it's useful for us to know what you're thinking.
Yeah. There's a collaborator in Japan working on the Anderson acceleration. We don't know if it's going to be ready by the time we need to start spinning up. So we'll start a root force, and if that's.
I was thinking less about that and more about like, given what you know so far about how the ocean is working, like, so if the oxygen minimum zones are looking better, like, what are you thinking that ocean BGC is going to look like when you spin it up?
Well, I hope it's going to be fairly stable. I mean, I mostly focus on the surface ocean phytoplankton, make sure those aren't drifting as much. But then also we have, the ocean deep circulation, making sure that we get enough upwelling in regions that where it can be sluggish, or like it was sluggish into a CSM2. And so, the backscatter parameterization should help in the equatorial Pacific, but I'm not sure that we're going to have as good of overturning in other areas like the North Pacific or the Southern Ocean. Now, we have seen that the bottom water cell is a little bit weak. CSM3 so far. I know that Gustavo and Ian have been trying to get that to be a little bit more vigorous, so that would help with all these like deep ocean circulation. But I think it's better than CSM2, but it's still a little bit weak. So hopefully, it doesn't cause huge problems, but I guess we'll find out, because that's we have to do a spin up to find out.
Related to spin up, I mean, the Anderson acceleration is maybe that'll work. And as you guys have pointed out more recently, it's not going to like cut it from, it's not going to be a 10x improvement. It'll be 3x maybe. So we're still going to have, in the long run, challenges with ocean spin up. And so are you aware of anybody who's working on, AI methods, or is that, have either of you, I guess I'm looking at you, to begin to think about that, or?
It's on the radar, but we haven't really devoted active neurons to it.
Maybe the other way of putting it is, if we were able to find some sort of source of money to support this, would you guys be keen or willing to support somebody who's gonna do the work?
Yeah, I mean, it would be nice to help to interpret, the results and that kind of thing. I'm not an AI specialist, but I could help provide the expertise.
Another one.
Sorry, and other people should definitely chime in if they have something. I just find it sort of interesting. We're seeing all these parameter ensembles on land, and it feels like the spin up is one of the big barriers to doing that in the ocean, because the timescales are so much longer. But it would be really neat if there would be some way to speed it up enough that, maybe not to the same extent, but that we could explore more of how the ocean BGC depends on these assumptions that we make. Really interesting.
That's absolutely right. It would be really neat. Keith and I have discussed this before, doing something similar, but we just don't have the resources right now. But we could, like you said, the timescales for like the full ocean carbon cycle would be that would be an issue. But for upper ocean, BGC and making sure we have phytoplankton distributions correct and nutrient concentrations, nutrients are really well observed. So in carbon, we could, you could try to match that part of the ocean. Let's assume that if we let the model go, then maybe the deep ocean would be okay too. But there's definitely a lot of observations. There's satellite observations of chlorophyll and primary productivity, and then all the ship-based measurements of all the other things. So it's definitely a possibility. It would be a cool idea.
We should probably break so that we can be back here at 11 for Rosie's seminar. So I think, again, all of our speakers, this is a great discussion.